Le agradezco mucho su presencia. Y bueno, yo solo voy a decir las palabras habituales que ya espero que todos sepan casi en memoria en cuanto a la seguridad, ¿no? En caso de que suene una alarma o haya un terremoto o algo así, tenemos que abandonar este auditorio de una forma ordenada. Entonces, pues ya saben, ¿no? La mitad del auditorio sale por esta puerta y la otra mitad sale por esta puerta. Ya esperamos que no pase nada y que podamos llegar al final tranquilos. También después de esto tenemos café y galletas por si quieren conversar con el doctor Josh Ross directamente. Y bueno, yo solo tenía que decirles eso. A la doctora Patricia Ornelas, de aquí del Instituto de Biología del Departamento de Zoología, va a ser la que va a introducir al ponente de hoy, ya que lo conoce mucho mejor de lo que yo. Y bueno, Pati, muchas gracias.
Bueno, para mí es un gusto presentar hoy al doctor Joshua Ross. Es un buen amigo y la verdad es que me siento muy contento de que haya aceptado venir a visitarnos en esta ocasión. El doctor Joshua Ross nos hizo favor la red de fauna nativa en ambientes antropizados de organizar un workshop, y derivado de este workshop fue que surgió la oportunidad de que viniera a darnos un seminario. Entonces, bueno, rápidamente para presentarlo, el doctor Joshua obtuvo la maestría por la Universidad de Denver, Colorado, en el 2001. Después fue estudiante de doctorado por la Universidad de Harvard y obtuvo el grado en el año 2005. Estuvo trabajando también en la unidad de medicina en la Universidad de Harvard hasta el 2009 y actualmente está dentro del departamento de biología en la Universidad de Cincinnati. Tiene revistas o artículos publicados en revistas de alto impacto con más de 25 contribuciones, entre las que destaco artículos en la revista Genetics, Nature Communications, Plus One, Heredity, Developmental Dynamics, entre otras. También ha tenido contribuciones en libros y capítulos y claramente dentro de su labor docente actualmente se encuentra activo en la Universidad de Cincinnati y ha formado doctores y maestros en ciencias en este tema. Para mí es un gusto porque finalmente el doctor Ross es una persona que ha trabajado muchísimo en el desarrollo de frontera en temas de transcriptomas y análisis de adaptación local en fauna de ambientes contrastantes. Entonces, sin mucho más, me da mucho gusto tenerlo aquí hoy y le agradezco. Thank you, Josh, for being here today. So, thank you for being here.
Thank you very much, Patty. Can people hear me? Is that okay? Okay. Thank you very much. I only understood about 2% of what Patty said, so I'm just going to trust that the rest of it was reasonably kind. In all seriousness, I want to start by thanking the Institute and UNAM from the bottom of my heart for the honor of coming and talking to you today about some of our work. Mexico is my home away from home. It is my favorite country to visit, and my Mexican colleagues earnestly are my favorite colleagues in all the world. So I always have such a wonderful time. It is always such a great pleasure for me to visit, and I thank all of you. And it's very nice to see some old friends in the audience today. So today what I'd like to talk to you about are some recent studies that we've done where we've looked at something called the transcriptome. So I want to start the talk by explaining what a transcriptome actually is. So when I talk about a transcriptome, formally what I'm referring to are all of the expressed genes and the level at which those genes are being expressed in a particular place in a particular time. So the term transcriptome is very general, and it can be very misleading. And so for the majority of my talk, what I'd like to do is try to qualify everything that I'm saying so that you'll be very clear that when I say a developmental transcriptome, I'm talking about all of the genes that have been expressed at a particular developmental time point. And I hope to convince you by the end of this talk that we've been able to utilize new sequencing technology to add insight into the nature of adaptation in the wild. And in particular, the animal that we look at, that my lab is dedicated to understanding more about, is the blind Mexican cavefish. So I imagine that many of you are familiar with this animal. This is a blind cavefish from the Pachon cave locality in an area that we call the Sierra de Alabra in northeastern Mexico. Near Ciudad Valles is the location of the village of Pachon, and the cave near that village is where each of these different animals are collected. This is the location. This is a very beautiful part of Mexico, and as you can see in this little northeastern sort of transect, you can see that there are a couple of different mountain ranges, and it's on the western edges of this principal mountain range where you see many different cave populations. So part of the reason why we study this system is because it's very valuable for understanding a spectrum of different important biological concepts. So part of the reason why we're able to utilize this very powerful system is because there are two different morphotypes. There's the surface morphotype, which I'm showing right here, that lives under a typical light-dark cycle. So this is just like any other sardine type fish that you may see living in nature. So a normal terrestrial or surface environment is going to have periods of light and dark, and the habitat of the particular surface dwelling form that we're interested in extends all the way through Mexico, parts of Central America, and up into the southern parts of the United States. So we can contrast that with the cave form, or the cave morphotype. So this is an organism that's closely related to this organism that is adapted to life in total and complete darkness most of the time. But I should say that there are some exceptions. These are distributed through several different populations, and the fact that they are relatively recent, a relatively recent divergence between each of these two different morphotypes still renders them capable of interbreeding. Because we can interbreed these individuals together, we can gain insight into the segregation of different traits utilizing classical techniques such as classical genetic partitioning, or looking at the inheritance patterns between of a particular surface or a particular cave dwelling trait. The spectrum that I talk about is very valuable. We can learn about the cave, we can learn about aspects of evolutionary biology, theoretical aspects such as regressive evolution, which I'll talk about in a second. As I said, we can use classical genetic breeding techniques to look at how and why traits differ between the cave and the surface dwelling form. There are a number of different behavioral differences that have evolved in cave dwelling forms. So unlike surface fish, for example, which show very strong coordinated movements and schooling behavior, the cave dwelling forms swim around tanks and swim around in pools in nature completely at random. So they have in essence lost their schooling and their shoaling behaviors. There are also a number of physiological differences. So the surface and the cave dwelling forms differ very much in their metabolism of oxygen. We've been able to utilize cave dwelling fish for a project where we look at facial asymmetry. Facial asymmetries are one of the most common birth defects that impact human beings around the world, and we know very little about the genetic basis for many of those the lack of coordination between the left and the right side of the face, the asymmetry. It turns out that cave-dwelling fish demonstrate robust and common, very frequent and dramatic types of facial asymmetry, and using genetic analyses we're able to gain insight into how and why facial asymmetry evolves in nature. There are also neuroscience differences, and what I'll talk about for the most part today is how our cavefish have now just recently, in the past year or so, entered into what we call the genomic age. So these cavefish, however, are not unique in the biological world. So irrespective of whether you are a vertebrate living in Mexico, or an invertebrate living in Slovenia, in Europe, in a tropical cave, or a temperate cave, if these animals colonize the cave, all of these animals tend to move in the same phenotypic direction. So there appears to be certain universal pressures related to the environment that drive animals in the direction of things such as losing pigmentation, losing vision, and enhancing non-visual sensory systems. We normally tend to regard these traits as each of two different categories. Regressive traits are those that are lost in the cave-dwelling forms, such as vision and pigmentation. Constructive phenotypic traits are those that become enhanced in the cave-dwelling form, such as the ability to find food and survey their environment. An important piece, I should say also, which adds to the tremendous power of this animal system, is the fact that we do not just find one cave population in nature. We actually find very, very many. So there are actually 29 different cave populations. Each one of these black spots represents a distinct cave population that's distributed throughout this relatively large region of the Sierra de Alhambra. And they are present in very different geographic formations, very different types of caves, and they're present in areas that actually have different watersheds. So the water that interacts with the caves over here is very different from the water that enters into the caves 2 valleys and 1 mountain range away. So what this tells us, what we believe this means, is that many of these cave populations actually invaded the caves at different times in the geologic past. And thanks to a lot of work that was performed by Dr. Patty, we actually know that we now regard there being sort of two distinct clusters. There's this red cluster, which we call the old or the ancient lineages. These were invaded by a surface-dwelling, a set or sets of surface-dwelling populations that became locally extinct and were pushed out to the Yucatan Peninsula, although there's some evidence that there are some of these ancient lineages still in the area, as well as a younger set of cave populations to the north and to the west as well. Interestingly, what we find is that irrespective of very different times in the geologic past when these cavefish enter the caves, they still converge on nearly identical phenotypes. So, what is the value of this system for evolutionary biology? Well, one of the reasons why I became interested in studying cavefish in the first place was that we actually know fairly little about a phenomenon called regressive evolution. So regressive phenotypic evolution is merely the loss or the diminution of traits in a derived lineage compared to the ancestral lineage. So if you compare a non-human primate to a human primate, what you tend to see is that we've lost the position of body hair, a good deal of changes to regions and quality of pigmentation. If you compare this animal to this animal right here, the ancestral form has a number of very robust limbs. This is actually a skink that is nearly limbless. So this is not a snake. It actually has very small limbs. Regressive evolution is also very nicely illustrated by horse digit evolution. So ancestral horses had multiple digits, which over time regressed until we reach modern day horses that in essence stand on a single digit. And regressive evolution has been an important question for a very long time. Charles Darwin himself discussed regressive evolution in The Origin of Species, where he said, specifically referring to cave animals, it is difficult to imagine that eyes, although useless, could in any way be harmless to animals living in complete darkness. He attributed their loss to disuse. So what are the principal theories that we think about when we think about regressive evolution these days? Well, the first theory is neutral mutation, and this is the idea that an animal, a surface-dwelling animal, may invade a cave, and the gene or genes that are responsible for making and maintaining an eye are no longer useful. And if they're no longer useful, then they're free to accumulate mutations and just drift out of the population. The second theory is selection, and one idea is that there is direct selection, which would argue that there is something actively harmful about trying to maintain an eye in complete darkness, and there, perhaps due to energy costs. And in order to conserve energy costs in the nutrient-poor environment of the cave, it may be that an eye is actually directly selected against through negative selection. A third theory is indirect selection, which argues that there may be some type of pleiotropic interaction wherein strong selection for one particular trait, perhaps a trait that's very constructive in the cave, actually impedes or somehow interferes with the normal development and maintenance of an eye. And in doing so, the eye is then lost indirectly by way of selection for some other related phenotype. Now the problem with all of these theories is that they hinge on our understanding of the gene or genes that govern these phenotypes. And the problem for many years in our field is that we didn't have very much in the way of genetic or genomic resources. So we decided to try to capitalize on a technique called next-generation sequencing, where we were able to accumulate a very large number of sequences, mass sequence them, and in essence try to build something that we call the transcriptome, or the set of all genes that were expressed at a particular time and place. So what I did a few years ago is I surveyed the entire literature to place this into perspective. So I should say that the cavefish were discovered in 1936 by a Mexican surveyor, and they were popularized by way of a commercial cavefish vendor, so someone who actually took these fish and put them and distributed them into pet stores worldwide. And I should say that the same lineages that were caught in 1936, we believe those to still be the same lineages that you get today if you go to an aquarium shop and you buy a live Mexican cavefish. In any case, much had happened in the first, say, 70 years of research into this system. So very early on, there were a number of classical genetic studies. However, we didn't know the gene that underlied those traits. A series of complementation or breeding studies indicated that two different eyeless cave populations are actually capable of producing offspring that do have eyes. So again, this hints at aspects of the genome that are changing differently in some of these different cave populations. But it really wasn't until 2013 that we were able to really dramatically enhance the number of actual gene sequences that we have available for this model system. And we performed this study starting in about 2011, where we took a male and a female surface fish and a male and a female cavefish, and we pooled their RNA together and we sequenced them to a very, very high depth. So we got over 1.5 million sequences utilizing a particular sequencing technology. And this enabled us to put together the first transcriptome analysis, which included over 21,000 new genes. And I should mention, at the time that we had completed this transcriptome, there were only 40 genes that had ever been sequenced in the history of biological research for this system. So utilizing this very powerful technique, we were able to really add a good deal of resource for further studies, which I'll tell you about today. What was interesting was when we made our transcriptome, we made it in an integrated way. So in other words, we took all of our sequencing reads that we were able to get from the surface fish, all of the sequencing reads that we were able to get from cavefish, and we essentially threw them together in a big pot. And we said, what are all of the genes that we can align together through using each of these two organisms? And it turned out to be a very large percentage, about 94% of these 21,000 genes that we were able to identify, 94% of them were built using information from both cave and surface. But what was interesting was that some of the genes that we were able to identify were only built using sequences from surface wellfish. And conversely, other genes were built only using sequences from the cavefish. So what this told us was that even though these are two relatively closely related animals, okay, there's some genes in the adult of surface fish that are constantly being expressed at reasonably high levels that are not expressed at all in cavefish. And conversely, the same holds true for cavefish. Cavefish are expressing very large numbers of certain genes that are completely absent from adult cavefish. So we wanted to find out what those genes were. And the way that we did that is we performed a series of filters, which I'm showing right here, and then we created a relatively solid list of all of the genes that are expressed exclusively in surface fish and not in cavefish. Somewhat not surprisingly, all of these genes were related to the visual system. So these are things like lens crystalline proteins, which are only expressed in a lens, things like visual system transcription factors, such as VSX1, and other eye structural proteins that are important for maintenance of things like the photoreceptors in the retina. So why this was interesting to us, and actually a nice proof of concept, is the fact that these fish don't actually have any eyes whatsoever. But imagine also that this provides some de facto evidence for the high energetic cost that goes into creating and maintaining an eye. So this may lend some support to the idea that the visual system is actually negatively selected against in the cave. So we next wanted to pursue the functions of these genes. And the way that we did this is we utilized a universal database that's called gene ontology. So I'll refer to this for the rest of the talk as GO terms. So whenever you hear the term GO, or GO term, what that's referring to is merely function. So for every gene that's ever been characterized in the human genome, based on a good deal of empirical laboratory research, as well as literature research, we know what those genes do. So if a gene is important for maintenance of the visual system, or oxygen respiration, or some type of particular behavior, then there will be a GO term that's associated with those genes. What you can do, a very nice technique, is you can actually survey large whole sets of genes, and you can say, what is overrepresented in this set compared to sort of a theoretical baseline. This is called a GO enrichment analysis. When we performed a GO enrichment analysis for these adult genes that were being expressed in surface fish, lo and behold, what we found were a large number of terms related to the visual system. So things like sensory perception, visual perception, structural constituents of the eye. All things that are reminiscent of the individual genes that I showed you in the previous set. So then we became very interested in asking what is happening with the cavefish. They don't have an eye. What are going to be sort of the obvious features that are going to fall out of an analysis of function for these exclusively expressed genes in cavefish? And interestingly, what we found were almost every one of the reduced GO terms were related to metabolism. So they were words like lipid metabolic process, oxoacid metabolic process, cellular ketone metabolic process, all processes indicating the function of the high level of expression of genes that are important for our alternative metabolic strategies for organisms that in essence are living under starvation conditions. Now what is interesting for us, or what we found interesting, is the fact that these individuals that we actually used were not starving. They were fed very, very well and under captive conditions in our laboratory. Fed 3 times a day, they were never starving. So what does this tell us about the underlying genetic basis? It argues, or provides some indirect evidence, that there are actually genetic changes that have evolved in the cavefish lineage that, in essence, causes the fish to believe that it is constantly living under starvation. And therefore, it is constantly churning out high levels of expressions of genes that are designed to help deal with starvation conditions. And I should say that this matches the ecology of most of the caves that we know about in the Alhambra region, where they are very, very sparse with food. But interestingly, if you go and visit these caves, you'll tend to find that the fish are fat and happy and swimming around very quickly. So from here, we wanted to take our work a step further. And the way that we did this was by performing developmental transcriptome profiling. And I'll tell you why we did that in just a second. But the 2 principal questions that we wanted to answer was, how does gene expression change over the course of development? And the second question that we wanted to ask was, we wanted to go beyond a single cave population, and we wanted to look at a second cave population. And the reason for this is that we wanted to ask the question of if evolution favors the same genetic, the same transcriptomic route every time an organism colonizes the cave environment. So in other words, are the same genes selected? Are we going to see the same genes being upregulated in cavefish in a population from the Alhambra as it is from a population from maybe one of the northern or western regions as well? So to what extent are independent cave populations adapting genetically to the cave environment in the same way? This was sort of the big question that we wanted to ask. And our studies came at a very good time because right when we began this developmental transcriptome project, it turns out that the cavefish genome had been published. So it is still not entirely complete. It's still regarded as being in a draft stage. But for our purposes, I should say that this genome project was able to identify, it's believed, no less than 98.5% of all genes that are expressed and present within the Cavefish genome. So this provided us a very powerful resource for starting this project. So what did we do? Well, we decided to survey across development each of 3 different types of fish. The surface-dwelling fish, this is what we would regard as the new lineage, or the current surface-dwelling forms that you find in the region. We surveyed cavefish from a locality called Pachon, and we surveyed cavefish from a more southern population called Tina. It's not important that you remember the names of each of these populations, just know that there were 2 what we believe to be independent origin cave populations that were surveyed. So, what is interesting is at face value, these 2 fish populations look identical to one another. So I would challenge anyone in the audience to look at each of these 2 fish and find anything that's obviously similar between the 2. But it turns out that if you look very closely, there are some more subtle differences. So for example, Pachon cavefish tend to demonstrate a little bit of yellowish pigmentation in their tissue. We think that this is related to an inability to metabolize certain types of carotenoid nutrients. So the Tinaha cavefish is a little bit more pale, so you can see this sort of looks clearish. This looks has a little bit of a yellowish or orangish tinge to it. Interestingly also, Pachon cavefish are completely albino, so they are formally albino, completely incapable of producing any melanin whatsoever. In contrast, if you look very, very closely, you can see little tiny speckles of melanin. So although Tinaha cavefish are depigmented, they are not formally albino. And so, as I said, we profiled across 4 important developmental stages. Those developmental stages are represented here by cartoons. So this is an egg 10 hours after it's been fertilized. This is an egg after 24 hours of development. You can just begin to see the eye and the tail sort of wrapped around the yolk sac. This is an organism at 36 hours post-fertilization, and this is an organism at about 3 days post-fertilization. All of our, we made very extra special care to make sure that all of our pools of developmental stages were identical. And so we had, in essence, the exact same developmental stages, and we profiled surface as well as each of 2 different cave populations, as opposed to 1, as it sort of appears right here. The other important piece to remember is that a lot is happening at this particular stage of development. So at this point during development, it is right about here that the eye begins to degenerate. So I should mention that very, very early on, there is a lens placode. A retina does begin to differentiate in cavefish, but after about 24 hours post-fertilization in Pachon, the eye begins to degenerate and sink into the orbit, and then is ultimately replaced with sort of fibrous tissue, and is completely absent, structurally absent from the adult. Also, pigmentation begins to regress during this developmental stage as well, so we wanted to capture what was happening over this dynamic developmental time period. So we performed 2 sets of comparisons. We compared the Pachon, or 1 cave population, against surface-dwelling fish, and we compared the other cave population, Tinaha, against surface-dwelling fish. So that's what's represented in each of these 2 schematics here. And again, we just surveyed across each of 1, 2, 3, 4 developmental stages. And in each one of those developmental stages, you can imagine that there are some genes that are going to be upregulated or expressed higher in cavefish relative to surface fish. Many genes that are going to be downregulated or expressed at lower levels in cave versus surface fish. And then those genes that are expressed at about the same level in both morphotypes. So this is where the matrix sort of begins to get a little bit complicated. But just know that there were 4 different developmental stages. We compared across all polarized ranges of expression, and there were 2 principal comparisons, each of 2 different cave populations back to surface. So what were our predictions going into this project? Well, I can tell you that we actually didn't really know what to expect. There was some argument that the same or similar genes may govern expression changes through cave colonization, because there's such a strong, as I showed you, similarity of external phenotype. The second reason is that we know that distinct populations So a population to the north, which is part of the young complex of caves, and the older cave complex. Each of those two cave populations will evolve albinism, and they actually do so through the exact same locus, OCA2, as well as representation of a trait called brown, which is governed by the same gene, MC1R. We also reason that selection may favor particular genes. So if natural selection is acting very strongly in the cave environment, then it may be that certain genes represent good targets, so to speak, for evolutionary change. But conversely, there was some evidence to suggest that we may find different genes being implicated in cave colonization for each of the two populations. And the first comes from complementation crosses. So again, these are just studies where you can take each of two different eyeless cave individuals from two different caves. You can breed them together, and their offspring will sometimes show the development of a relatively normal, we believe sometimes functional eye. So what that means is that different genes must be, at least some different genes, must be governing the loss of eyes in each of those two different cave populations. We also know, one argument that we may see a large number of different gene expression patterns comes from the work of Patty, Martina Braddock, and others, that has shown that there's a very distinct evolutionary history across the cavefish populations. And by geography and time alone, one might expect that randomly we would see different gene expressions. As well as the fact that, as I showed you in the previous slide, there are apparent differences in the phenotype. So in terms of the patterns of gene expression, regulation, what did we find? Well, this was perhaps one of the most surprising things. This was one of the simplest pieces of data to pull out of our data set. And what we found was, whether you're looking at the first developmental time period, the second, third, or fourth, interestingly, the numbers of genes that are up or down regulated in each of our two cave populations is very, very different. So comparing to surface-dwelling fish, for example, there are many more genes that are underexpressed in Tanaha than there are in the Patjone cave population at 10 hours post-fertilization. And conversely, there are many more genes that are overexpressed in Patjone than there are in Tanaha. And you can see that this pattern changes throughout each of the 4 developmental periods that we looked at. So our first clue is that, in fact, no, it's going to be very, very different changes are occurring in each of these two different independent cave populations as they had adapted to the underground environment. So we next move to surveying go terms. So again, we're looking at function and asking what are the functions of genes that are being enriched in each of these two different cave populations. And the way that we did this is we tried to get a handle on a very, very large data set. So we were dealing with nearly 1 trillion 50 base pair sequences at this point, and we were dealing with a very, very large genome, and we were dealing with many, many individuals. And so in order to try to get a handle to simplify this data set, what we did is we worked under the assumption that the genes that are the most differentially expressed, that is in the top 2% highest differences between Patjone cavefish and surface fish, would presumably be genes of some type of ecological relevance. Now this is an assumption, but we think that it's an informed assumption because it would argue that there wouldn't be such a dramatic difference in gene expression if there was no purpose behind it. At least this was our assumption. And so when we did that, what we found was each of 3 different categories. We found that there were some functional terms in that rarefied category that were specific to Patjone. So these are related to things like pheromones, the ability to detect one another, detection of chemical stimuli, so the ability to find taste mints. Interestingly, there were also go terms that were unique to the other cave population. In Tanaha, we found things related to the storage of lipids, the ability to deal with a heightened immune system in the cave. But we also found that there were several go term categories that were shared between each of the 2 different cave populations. And so these are sort of universal things like sensory perception of taste and immune system processing and things related to antigen presentation and so forth. So we found that very interesting. There's a hybrid, so to speak, of functional categories that are being enriched, but they're being enriched in different ways depending on the cave where the fish is located. So going back to this category right here, we said, okay, well, sensory perception of taste this is the same Go term is popping up in each of these two different categories, these really overexpressed genes in each of the two cave populations independently. So we said, well, is it the same genes that are reporting that functional category? I should say that there are three possibilities. It could be that the exact same genes are reporting sensory perception of taste in each of the two cave populations. It could be that they're entirely separate genes. Or the third category is that it may be some mixture of the two, shared and different genes between both cave populations. And lo and behold, what we found was that it was the third category. So there, when you look at all of the genes that are responsible for that heightened functional category of the ability to taste very well in the darkness of the cave, one gene has been selected for independently in Pachon, two genes have been selected for independently in Tanaha, but there was one gene, a taste receptor, that has been heightened in expression in both of the cave populations at one particular developmental time. So we found this very interesting. Even in cases where the same functional categories are being selected as a consequence of life in total darkness, it's not always the same genes that are mediating that process. So we found a very similar theme when we look at genes that are downregulated or underexpressed in the cave population. So in Pachon, there were exclusive terms relating to negative regulation of appetite, eye pigmentation, those that were exclusive to the Tanaha cave, including sensory perception of smell and lens differentiation. So keep in mind that these categories are not coming up in Pachon. So only Tanaha is showing genes that are important in this category for the sensory perception of smell. But again, we found certain categories that seem to be universal or important to both cave populations, including, and interestingly, the downregulation of genes important for absorbing visible light, and genes that are important for photoreceptor activity. So a very intuitive result, as you might expect for animals that did not have vision. So we next turned our attention to specific genes, and we wanted to ask, if you look in profile, just individual genes, rather than functional categories, rather than looking at Go terms, if you survey particular genes, do we see this type of pattern? And in fact, we do. So here's one example of a category that we call convergent reduced, where you have a gene called SOL4. This is a gene that's important for digestion. It's expressed at very, very high levels in surface-dwelling fish across all 4 developmental stages, and it's downregulated to near 0, to near nothing, in each of 2 different independent cave populations. Conversely, we found genes that are convergently increased in their expression. So look at this gene right here related to keratin metabolism. It is expressed at higher levels in each of 2 cave populations and nearly baseline in the surface-dwelling population. And again, this is across each of the 4 developmental stages, the dynamic stages of development that we surveyed. So we then said, okay, well, let's see if we can validate some of these transcriptomic analyses. This is working with very, very big data, and it's rife with concern around false negatives, false positives. So we said, well, let's go through and see if we can validate. And in fact, what we found was that for each of our categories, whether convergent increased, convergent decreased, divergent increased, or divergent decreased, what we found was that our patterns of RNA sequencing, which are shown right here, where you would have a high level expression, say in surface and Tanaha, but no expression in Pachon, you can see the exact same result is recapitulated very nicely with qualitative PCR. I should mention this is a gene strongly expressed in surface and Tanaha, absent from Pachon. And down here I'm just showing a control gene, which is showing that we're able to find normalized expression across all 3 crowns. We also found sort of the opposite situation. So this is an uncharacterized gene called CB1081. This gene is expressed at very high levels in Pachon, but completely absent in surface and Tanaha based on our RNA sequencing analyses. And in fact, you can see we only have one strong band when we perform qualitative PCR, exactly as we would expect for validation. So back to the genes. won't bore you with the details, but the fact is we were able to find many genes that demonstrate both convergent and divergent levels of expression. And so if we just take the top gene right here, this is a gene at 72 hours post-fertilization. Again, we call this convergent reduced. So in both cave populations, this gene has been reduced to almost nothing. And interestingly, it's a homeobox visual system gene called BSX1. The GOAT term is visual system, and you can see that it is very dramatically downregulated in each of our two different cave populations. So something, again, intuitively that we might expect. So the nice thing is that we found both patterns of convergence, okay, where you see down, 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 up, 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 but we also found very divergent patterns of expression. So again, it seems to be a mixture. It's not always the same genes, and it's not always the same polarity of gene expression change. So here, just using one example, the gene AGRP, which is a very important gene for pigmentation, it is expressed at very, very low levels in the Patchone cave, but it's actually expressed a little bit marginally higher in the Tanaha cave than it is in the surface cone. Excuse me, in the surface cone. And interestingly, these patterns of convergence and divergence were not uniform across development. Very different genes popped up at each of the 4 different developmental stages. So when we went through and surveyed the genes that were important at 24 hours post-fertilization, we nearly wiped the slate clean, and found a very different set of genes that were up and down regulated at 24 hours post-fertilization. And so here I'm just showing you some examples again of genes that are convergent, reduced, and increased, and those that show very divergent results, where they may be very high in Patchone, but show the exact same level of expression in surface fish. And again, this is our 3rd developmental time point at 36 hours post-fertilization, and this is our final stage at 3 days post-fertilization, where again, we're finding fewer and fewer genes that seem to be important for visual system, and many more genes that become sort of divergent in their patterns of expression. So it almost indicates that as both cave populations move through development, the older they get, the more their genes seem to show a lot of differences, minor or major differences between them. So I'll finish by telling you about the final experiment that we performed, which is something called an ingenuity pathway analysis. So pathway analysis, in essence, is a way of taking your massive data set and trying to simplify it. And the way that it is simplified is by looking at the coordinated patterns of expression for many, many genes. Okay, so you notice that things got very complicated very quickly. We had many genes that were going up, many genes that were going down, and they seem to be very different between the cave populations. What this analysis does is it looks sort of holistically at the entire landscape of the transcriptome, and it looks at all of the different patterns of genes that are being expressed and the way in which they're being expressed. And it predicts, or it says, I think that this gene is probably downregulated based on the fact that all of these other genes that it regulates are similarly decreased or increased in their patterns of expression. Okay? So when we did that, we were in essence searching for sort of master regulators, or sort of important upstream genes that may play sort of key roles in cave colonization. And what we found was something very, very exciting, which was when we performed this analysis in Patchone cavefish, and when we performed it at one particular developmental stage, at 36 hours post-fertilization, what we found was a large number of genes related to both pigmentation and vision all seem to point their finger at one transcription factor called OTX2, which we know from prior experiments to be downregulated in Patchone cavefish. Interestingly, when we performed this exact same pathway analysis in our other cave population in Tanaha, it nominated the exact same gene. So, even though there's a good deal of noise, there's a good deal of things that are changing throughout the entire transcriptome, there are certain core genes that are changing in a very coordinated way that seem to suggest that in each of these two independent cave populations, OTX2 seems to be very, very important. for both pigmentation and vision loss. It didn't stop there though. There was one other gene that we were able to find, which I found really exciting as well. So again, in patron cavefish at 36 hours post-fertilization, based on a much larger set of genes that seem to be co-regulated in a particular pattern, the pathway analysis predicted one gene called MID-F. This is a gene that stands for microophthalmia transcription factor. This is a transcription factor, again, that plays key roles in vision and pigmentation loss. It came up as a very significant hit. Again, when we did the same analysis in Tanaha cavefish, we found the exact same gene. So, in conclusion, what we think this tells us is that from a broader perspective, very diverse gene expression changes can underlie adaptation to extreme environments. And so the evolution of very, very similar traits, and using cave animals as just one example, this seems to argue, we believe, that very, very diverse changes can sort of underlie that, the evolution of those phenotypes. Genes that are expressed in very similar ways and those that are expressed in very different ways. We interpret the shared or the convergent gene expression changes as those changes that sort of reflect the global pressures, the most important proximate pressures that are probably impacting animals, in our case, that invade the complete darkness of the cave. So they need to deal with starvation, they need to deal with the fact that there is no photosynthetic base, they need to deal with the fact that there is no light. Unique were some of these divergent transcriptomic changes, which we found many of which that we found in our data sets, probably, we believe, represent or reflect subtle changes that are more specific to the individual ecological environments, the specific caves where each of these different cave populations have evolved. I should also mention that some of the transcriptomic changes that we saw may also reflect recent or periodic hybridization back with surface dwelling fish, but we saw less evidence of that. We also find that certain master, quote-unquote master genes like OTX2 in MID-F appear to be very common targets for cave adaptation, and convergence, and in some cases parallelism, can be detected at many different levels of analysis. So keep in mind that one can assign selection perhaps at the level of function, gene function, but that gene function may not always indicate or may not always reflect the underlying same genes that are governing those gene functions. And so I'd like to end there, and I'd like to acknowledge funding from the National Institutes of Health as well as the National Science Foundation. I have a great team right now of a few different graduate students, research techs and undergraduates, as well as excellent collaborators, one in particular right here, as well as my graduate student, Bethany Stahl, who led the majority of this transcriptomic project. So I'd like to thank you for your time, and if you have any questions, I'd be happy to include them.
Tenemos tiempo para un par de preguntas, si quieren hacerlas en español, tengan aquí Pati y yo intentamos traducirlas. Oscar.
Have you considered now going to study some of the blind catfish and see if the same master genes are affecting the progomorphisms?
Right. I would love to do that. I think right now we invested so much financially into the resources. I feel somewhat beholden to keep going with Astyanax for a little while. But yeah, I think that if we can get our hands on some blind catfishes, it would be phenomenal. Yeah, we would love to do that. And I think that one of the nice things about this type of technology is it really puts into anyone's hands the ability to perform whole-scale big data projects like this. So it's no longer the case that you need to have a genome, an entire sequence genome. We now have the ability to perform de novo analyses. So it's probably very much within the realm of possibility to look and see if there's convergence across much deeper phylogenetic differences such as the catfish.
If you discarded the possibility of gene flow between these two cave populations on the surface, because this factor could introduce a lot of noise in the analysis.
Yeah, so that's an excellent question. So the question was, did we consider the possibility of gene flow occurring between the caves as well as between the cave and the surface? And we did. So we know about gene flow that has occurred even just in reports, natural history reports going back to the 1960s. We also know that the phenotypes within the caves have changed very dramatically in the same time period. So I talked about Pachon, formerly being albino. In the 1960s, it was not an albino population, at least not exclusively. So there was some albinism, but there were some individuals that were pigmented. By the 1980s, interestingly, the number of individuals who were pigmented actually went up. And then by early 2000, the number had dropped to nothing, so that as far as I know, no one has been able to identify any fish from Pachon that are anything other than albino. And so the phenotypes can change very, very dramatically, very quickly within the cave. And some of that I attribute to introgression with the surface-dwelling forms. And so, you know, in terms of the, I should say, in terms of the noise that we see, some of the details of our methodology controlled a little bit for that. So we used sort of purebred, what we regard as purebred cave populations that have been reared, inbred within within captivity for at least 7 generations for both cave and surface. And so we think that that reduced some of the noise within sort of the population of individuals that we were pooling. But having said that, the question that you get at is a very good one, and it's very well taken, and I think that we would need some alternative source of information to really get a handle on the extent to which some gene expression is actually has been influenced by recent introgression with surface fish. I don't know how to do that. It's a very good question.
George, thank you very much for the talk. And I was wondering, this hypothesis about some master genes are the same in Pachon or Tinaja, and other, like Ocados or other pigmentation genes, could be influenced by maybe interbreed on the surface conditions. What do you think about these master genes that are actually, despite there could be introgression, are still getting selected in these kinds of phenotypes, even when we're maybe outside of the natural environments? How do you feel this could be the case in this, or in other populations? Like there are several caves that has been proved to be interbreeding between the surface and the populations.
Right. And so, I'm sorry, so your question is how, like why is OCA2, for example, not a master regulator, or?
What I maybe mean is like, if you have this master regulator like
OTX, yeah.
involved in Pachon and in
Right.
Tinaja, what is about these genes that actually decides that there could be integration with the surface fish? They can continue being in this same direction.
I see.
And you are not lost in this.
I
Or
see.
maybe in the hybrids you actually lost, or you interbreeding, it's making these genes to lose it.
Yeah.
Integration with the cavefish.
Yeah. Okay. So that's a very good question. So in other words, why is it that even in the face of some periodic interbreeding with surface-dwelling forms, why is it that OTX2, MID-F1 seem to be these master regulators that are able to remain static, they're able to sort of remain solid within the cave? Yeah, it's a good question. I guess, so theoretically one might presume that there's something advantageous. So if there is a very strong selective advantage perhaps to having the types of alterations to OTX2 and MID-F1 that we saw, if there is, then perhaps that selective value outweighs whatever influence would come from periodic introgression from the surface-dwelling form. That's one theory. An alternative could be that they're very, very old mutations. And so it could be, and this would be very controversial if I were to say this at the academic community meeting, but it may be that there is a very, very ancient ancestor, proto-ancestor, that seeded both the Pachon and the Tinaja cave. And it may be that OTX2 and MID-F1 sort of reflect that very, very early colonization. And so if and therefore if it was fixed within the cave population, one might one might then expect that it would change much more slowly, as you said, even in the face of periodic conversation. Yeah?
So at some point during your introduction, I think you mentioned that differential expression of I-related genes between the surface and the cave suggested negative selection of those.
Perhaps.
Why is that? I mean, why do you think so?
Yeah, well, so I should clarify and say, I don't know the answer. I don't know if it's negative selection or if it's neutral mutation. To give you a little bit of background, neutral mutation was really the dominant theory for many, many decades in cavefish research. And the problem that I always had with neutral mutation was that at the time, the belief was that cavefish were very, very young, that they had only colonized the caves since, say, the recession of the last ice age, so only about 20,000 generations or something in that arena. And if the argument was neutral mutation, that argument hinges on a great deal of time. So a good amount of time for, say, loss of function mutations to accumulate in these important genes for, say, pigmentation or vision. So I didn't like neutral mutation at first, but then Patty's work threw a big wrench into the whole project, or the whole paradigm, because we actually learned from Patty's population genetic studies that in fact the caves were probably colonized way, way earlier. And so the revision now, the consensus view, is that the cave populations are probably between 1 and 5 million years old. And so I think that enters the possibility now of neutral mutation as being at least a contributor to phenotypic loss. But based on the nature of a genetic lesion alone, I don't know if we can make that determination. Because I think that sometimes it can be attractive to think, well, here's a loss of function mutation that we identified, we discovered in a pigmentation gene, and it's fixed, and it has a really robust phenotype. So it's intriguing or attractive to say, well, that's probably neutral mutation because it represents the accumulation of a deleterious, a functional mutation to the gene. But the problem is that I don't think we can rule out the idea of negative selection also. So it could be that if that was a very rare deleterious mutation that popped up infrequently or rarely within the population, it could have spread very quickly. So I'm sorry for the long-winded answer. I think to get back around to your question, I think the best determination is to first identify what are the actual genes that are changing, what are the genes that are responsible for the phenotype, and sort of stop there. And then at sort of a genomic level, then begin to scan the genome and try to determine if there are any signatures of selection. And we can do that. We can look across the genome and see that there's a lower level of variation than we would expect by chance alone, or which would rule out the idea of neutral mutation. And I think that along with knowledge of the genes might, and perhaps some functional studies as well, may give us a better understanding of sort of the mechanism of evolution, which is, I think, what you're thinking.
And one a quick thing is when you did the transcriptions, were they all males or females? Or?
Yeah, so when we initially did our first one, our first analysis was one male, one female, because we actually never followed up on this, but we wanted to look at gender-specific, male or female-specific gene expression in each of 2 different axes. For the developmental transcriptome, it was too early to determine the sex of those individuals. And I should also mention, we had to, at the time, because of the technology, we had to pool 50 individuals together to get enough RNA to actually perform the technique. So it's most likely, we believe most likely a mix.
One more question.
What are the phylogenetic relationships, the 3 populations, Machón, Tinaja, and surface?
What's the phylogenetic relationship? Good question. I wish I could defer to my colleague. So I can give you my understanding, which is that we believe that Machón and Tinaja, they're both regarded as members of the same species, Astyanax mexicanus, at least by most people. there's some controversy. We believe that Pachon and Tinaja are independent cave colonizations, but they both seeded the caves from the same general population of the older surface dwelling form. Okay, so in other words, there's an older surface dwelling form that is no longer present in that region, and an independent subpopulation of that group colonized Tinaja, and a separate independent group colonized Pachon, probably at different times in the geologic past, but we don't know for certain when. The surface dwelling forms that we used are Astyanax mexicanus, and they are relatives of the surface dwelling forms that inhabited the younger caves to the north and to the west. Somewhat complicated.
Thank you very much.
Yeah, thank you. Thank you very much.